Alright folks, let's go over what you need to study for your test on genetics and heredity. In your VIP section, we have two different foldables from this unit. We did this brief intro to genetics and intro to heredity foldable before winter break. And then after winter break, we did a more complicated Punnett square how-to foldable. Also in VIP, you have a formative that we did with a gerbil or maybe hamster Punnett squares. We did quite a few lab activities this unit. You could look in your lab section for your genetics introductory web quest, your DNA dogs worksheet, your coin flip family, your Punnett square eggs paper. If you grabbed them, the SpongeBob or Harry Potter practice problem sets, the answers to these worked problem sets can be found on my Phoenix website. And from our review day, you have one or both of the gizmo sheets. And of course, you have your portfolio paper for this unit that you are working on, and as always, your glossary. Additional resources, including some awesome online review games, can be found by going to my Phoenix website. If you log into Phoenix Parent View or Student View and click on class websites, you can find full copies of the notes as well as worked problem sets and blank note sheets on the tab Homework and What Do I Need to Study. Review games can be found under Review Games, and links to this and other videos can be found under Miss E's videos. For as long as there have been people have wondered how traits pass from one generation to the next. Long ago, long before Watson and Crick and Rosalind Franklin, humans used what they knew about the genetic code, even though they didn't know what DNA looked like or how it replicated, to get desired traits. They used a process called selective breeding, where they would breed desirable traits in one animal or plant, in the case of crops, over and over and over through generations until they got the desired result. The domesticated dog was selectively bred from wolf pups. But it wasn't until the first half of the 19th century that people really started to try to figure out what was being passed between generations. So our father of genetics that we learned about in this unit is an Austrian monk named Gregor Mendel, who loved to garden. And Mendel had in his garden both tall and short pea plants, plants that had red flowers, plants that had white flowers, plants that had pink flowers, plants that had flowers at the top of their stems, plants that had flowers on the side, plants that made yellow peas, green peas, wrinkled peas, smooth peas, and everything in between. And Mendel took very detailed notes, and he crossbred lots of different kinds of plants to see the results. He crossed two very tall pea plants, and the resulting offspring were all tall. He repeated this experiment with two very short pea plants, and the resulting offspring were all short. He then took a short offspring from the short generation and a tall offspring and crossed them. When I say crossed, I mean made them mate by using pollen from one plant and putting it on the other. When he crossed the short plants and the tall plants, he was expecting that maybe some medium-sized plants would result, but that was not the case. What happened instead is that all of the offspring of this cross were tall. This surprised Gregor Mendel a lot, so he took two of the offspring plants from this new tall generation and crossed them. The results were surprising. There was a mix of tall and short plants in this offspring generation. He was observing the effect of recessive and dominant traits, although he didn't call them traits at the time, he called them factors. Mendel coined these terms dominant and recessive to describe what he was observing in his plants. He noticed that whenever a plant inherited the tall factor from a tall parent, it would be tall. The only way to see short offspring would be if they got this recessive trait. Because of Mendel's careful observations and his peas, he is said to be the father of genetics, and much of modern-day genetics and heredity studies are based on his findings. Years later, another budding geneticist popped on the scene. His name was Reginald Punnett, and he created a system known as the Punnett square that can be used to observe Mendel's inheritance rules in everyday life. We did a lot with Punnett squares in this unit. It relies on the fact that there's an equal opportunity for any of the gametes or reproductive cells from the parents, which carry the chromosomes, to join. And you can predict the offspring generation using your rules of probability in the Punnett square. Let's do a practice Punnett square problem. All of the Punnett squares that we did in class follow Mendelian inheritance, which is to say that they have complete dominance. That means that your dominant trait is always going to beat the recessive trait. So any organism that has at least one dominant allele is going to express that dominant phenotype. You'll only see the recessive phenotype if 2 copies of the recessive allele are inherited. Alleles, you'll recall, are different versions of genes. Phenotype, you'll remember, is the observable physical appearance or behavior of an organism, whereas genotype are the actual genes, the code that that organism has in its cells. Let's do a practice Punnett square about cats, my favorite. Here we have 2 parent cats. They have some similarities and some definite differences because no 2 organisms have the exact same genotype or genetic code unless they're identical twins, or the exact same phenotype, physical appearance. Let's let the letter capital G represent the dominant trait for gray fur. Let's let the lowercase g represent brown fur, a recessive allele. Now, neither of these parents have brown fur that we can see with our eyes in their phenotype, but it's possible that they're carriers of this recessive trait, this recessive allele for brown fur. Let's say that both of these parents are hybrids or heterozygous. That means that they're each carrying 1 dominant and 1 recessive trait. They'll both have the genotype capital G, lowercase g, a heterozygous hybrid genotype. Let's say that this parent's alleles are on the top, and this parent's alleles are on the left side. I will use 2 different colors so you can see all of the possible combinations of parent alleles which are on their gametes to see the possibilities for offspring. When I fill in a Punnett square, I want to take whatever letters at the top and whatever letters on the left side and draw them in. Those are going to be my genotype letters for each square. In our first square, we see a homozygous dominant or purebred for the dominant trait genotype. Whenever I have both dominant and recessive alleles, I'm always going to write the dominant or capital letter first. Here we see a heterozygous or hybrid genotype. Here we have another heterozygous or hybrid genotype. And last but certainly not least, we have a homozygous recessive 2 lowercase g's genotype. So we end up having quite a variety of possibilities here. We have some possible offspring that are purebred for the dominant trait of gray fur. We have some that are hybrid heterozygous for these alleles, and 1 possibility of homozygous recessive, 2 lowercase g's. What this might look like in terms of phenotype would be babies that have gray fur 3 times out of 4, and then 1 time out of 4, a possible offspring with brown fur. Now, this does not mean that these 2 cats will have 4 babies. It also does not mean that every time they have babies, it will be exactly this distribution. It just means that each of their offspring has a 1 out of 4 chance of having brown fur. Let's look at these ratios in a little bit more depth. We saw before that 3 out of 4, or 75% of the offspring, would have gray fur. We also saw that 1 out of 4, or 25% of the offspring, would have brown fur. Notice that there are 2 different genotypes, both homozygous dominant and heterozygous, that gave us this gray fur phenotype. This proves to us that we cannot tell by looking at something what its genotype is. We had 1 out of 4, or 25% of the possible offspring having this homozygous dominant genotype. We had 2 out of 4, or 1/2, or 50% with the hybrid genotype, and 1 out of 4, or 25%, with the homozygous recessive genotype. When you're doing these Punnett square problems, you want all of your percentages to add up to 100, obviously. That's how you know you did it right. As I said earlier, this is illustrating Mendelian inheritance with complete dominance. That means that your dominant trait is always going to win over your recessive trait. But it's not always the case in real life. This is not always how things work. Sometimes we see a case called incomplete dominance. This happens when perhaps we cross red flowers with white flowers and get pink flowers. This is a blending of traits. Other times we might see multiple alleles at work. Multiple alleles simply means that there's more than one place on a chromosome, or perhaps more than one chromosome carrying more than one gene that dictate a certain phenotype. We know this to be true just by looking around our classroom or looking at a picture of our friends. No 2 people have exactly the same eye color, skin color, or hair color. In humans, there's a great variety. That's an example of some traits that are dictated by multiple alleles. Now, all of these traits we've talked about are inherited traits, meaning that they're coded in our genes. I inherited some of my traits from my mom and from my dad. My mom and my dad got their traits from their parents. So here are pictures of my two grandmothers, mom's mom and dad's mom. I have in my DNA combinations of traits from all of these people, including my two grandfathers that I do not have pictured here. But what about the traits that describe me that are not in my DNA? Where did those come from? Like, was I born really loving to watch and play baseball? Nope. That's something that I acquired from hanging out with my dad. What about my love of singing? I learned that from my dad's mom, my paternal grandmother. My interest in crafts and sewing, learned that from my mom. My love of swimming, I can thank my mom's mom for that. She taught me my first swim lessons. My love of Latin and cats, that I guess I just learned at school or from somewhere. Either way, all of these things are acquired from my environment, either through learning or other experiences. Other acquired traits might be modifications to someone's appearance beyond what's coded for in their DNA. Like if I gave myself a haircut, or completely changing my hair color, or got a tattoo. So if it's an acquired trait, it's something that you learn through experiences. If it's an inherited trait, it's on your DNA. DNA, when it's condensed, is chromatin, and when it condenses even more, is a chromosome. We met these chromosomes for the first time in our cell cycle unit. But all of these chromosomes contain the DNA information, the code that make all living things what they are. These traits, these inherited traits, are passed down from generation to generation because this DNA is copied. This DNA is copied in the cell cycle. Specifically, the DNA that we all have in our cells came from our parents. It came from gametes, or reproductive cells, that are formed in the process of meiosis. Meiosis, you'll remember, takes one cell and splits it into 4 unique gametes, each with a half amount of information. Kind of like our eggs, when we did our egg activity, how each half came together to make one whole egg. To make one organism like a human, we've got half of the DNA from one parent, half of the DNA from another. The gametes fuse together to make a unique organism. It probably weirds you out to think about parental gametes, but that's what we're actually representing every time we do a Punnett square. Each time we set up a Punnett square, we're showing all of the possible combinations of the gametes or reproduction cells in the offspring generation. So when this whole cell cycle is happening, this whole big circle of life, where we're copying our DNA, when all of this is happening, sometimes stuff goes wrong. Just like when you're texting a friend or typing up a paper, sometimes you make a typo. When we have these typos happen, this is called mutation. Now, mutation and mutations get a really, really bad reputation, which is kind of unfair. Sometimes we think of them causing superheroes. Other times we think of mutations causing some kind of terrible, ugly disaster. But in reality, a mutation is just a change in DNA. The word mutation comes from the Latin word that means I change. The mutation is just a change. This can lead to evolution, which is our next unit. Let's go back to our cats example. Here we see that in any group of a species, there's some variety. There's also a lot of that species. Depending on the environment and other factors at work, a mutation or a change in DNA could be helpful, harmful, or neutral. Let's look at a case where a mutation might be positive, beneficial, or helpful. So you'll notice that some of these have gray fur. If they're living in an environment where gray fur might provide a kind of camouflage, like against this gray background, that might be a beneficial mutation to have because it might help them blend in and stalk their prey more efficiently. So in this case, having a gray mutation in the fur would be very helpful. In the same population of cats living in the same environment, it might be negative or harmful to have a fur color that makes you stand out. It might make you more likely to be noticed by predators or prey alike. So in this case, having a mutation for yellow fur or brown fur might be negative in this certain situation. Now, hopefully most cats aren't having to eke out an existence on the streets hunting for mice. Hopefully most of these cats are adopted and living in loving homes. In that case, their fur color doesn't matter, and it's a neutral or no effect adaptation. So all this stuff we've talked about, even going back to Mendel and his peas and Reginald Punnett and his Punnett squares, all leads us up to modern technology and modern applications of heredity and genetics. One of the most exciting things that we can do, knowing about heredity, genetics, and patterns of inheritance, is something called genetic engineering. Genetic engineering allows us to take advantage of the fact that all living things have the same 4-letter code of DNA. Doesn't matter if they're eukaryotes, prokaryotes, doesn't matter. They all have the same 4 letters. That allows us to take genes of interest from one organism, such as a prokaryote that can glow in the dark, and put them into another organism of interest, like a cat. Much like you would copy and paste on your computer, you can kind of copy and paste genes of interest from one organism to another using enzymes and create a new organism with the combination of desired traits. This is a real thing, by the way. Glow-in-the-dark cats, real thing. Except they use jellyfish DNA. Now's the part where I'm going to review everything
really fast. In this unit, we learned about 2 scientists. Chiefly, we learned about Gregor Mendel, who is considered the father of genetics because of his study with pea plants and their inherited traits. He coined the terms recessive and dominant, and he called them factors. Today, we know them as alleles. We also learned about Reginald Punnett, who invented, you guessed it, the Punnett square. All of the genetics problems that we did in this unit were situations of complete dominance. This is the case when the recessive trait is completely masked by the dominant trait, unless no dominant trait is inherited. We also discussed that it's not always as simple as just one trait masking the other. Often, there are many traits at work. We also discussed the difference between genotype, the genes an organism has, and phenotype, its appearance or behavior. An example of genotype would be capital R, lowercase r. An example of phenotype might be red hair. Additionally, we learned the terms homozygous, heterozygous, and dominant and recessive. Homozygous dominant organisms have all capital letters in their genotype. Heterozygous organisms have capital and lowercase letters in their genotype. And homozygous recessive organisms have all lowercase letters in their genotype. Another way of saying homozygous is purebred. And another way of saying heterozygous is hybrid. And just for fun, let's do a really sped up Punnett square practice problem. Let's imagine that in a certain species of frogs, the allele for green skin is dominant to the allele for a plaid pattern skin. Remember, this is just pretend. Imagine 2 green hybrid frogs had babies. Let's make a Punnett square to figure out what the possibilities would be of the babies inheriting various genotypes and phenotypes. Because each parent is hybrid, or heterozygous, in this example, each parent has one dominant and one recessive allele to contribute to the possible offspring. I'm now going to fill in the Punnett square to see what the possible genotypes of the offspring would be. I filled this in, this first row, by looking to see what's above and what's to the side, like this, to fill in the traits. I'll do the same thing for the second row. We end up having capital G homozygous dominant, 2 capital G lowercase g, or hybrid, heterozygous possibilities, and 1 homozygous recessive, all lowercase letters. Remembering that the dominant allele, capital G, represents the trait for green skin, we see that there is a 1, 2, 3. 3 out of 4 are 75% chance of the offspring inheriting the green skin phenotype, and a 1 out of 4, or 25% chance of having the plaid phenotype. The babies might look something like this.